At the beginning of my presidency, I asked Robert Langer whether he'd be willing to do the President's Lecture. We normally have a President's Lecture every year, and we've had to cancel him 4 times. And graciously, he's agreed to continue, to do the President's Lecture. He was going to come across last year, and then again later last year, and so on. But every time we've had to cancel him, and so on. And I was particularly pleased to get Robert Langer to be able to do the President's Lecture, because Robert Langer is perhaps one of the most important scientists in the world. And let me just read you his biography. He's one of 12 institute professors at the Massachusetts Institute of Technology, MIT. That's the highest honor that can be awarded by MIT. He has written over 1,500 articles. He's been cited over 348,000 times. And his H-index is 291, right? Somebody was talking about H-index and said that if you manage to get an H-index which is similar to your age, you're doing very well. So his H-index is 291, and that is the 4th highest H-index ever in the world. So he's got 400 patents, over 400 patents. He's co-founder of a number of companies, including Moderna. And Moderna has obviously become very famous. He's won all sorts of prizes, including the equivalent of the Nobel Prize for Engineering, which is called the Charles Stark Draper Prize. So Robert certainly is one of the most important people that we're going to have to be able to do the President's Lecture. And I was particularly glad that he agreed to do this when I contacted him, because I saw his lecture when I was in the States, in Boston, a few years ago. So he's going to tell us about our quest to solve global health challenges by developing new pharmaceutical technology. He's recorded this talk, and that's going to come on, and then he's going to sort of join us for live question and answer session. Thank you very much.
Well, first, let me start with how I got started. And it was kind of an unusual way to get started, because I'm a chemical engineer. And when I got my degree in chemical engineering, very few chemical engineers worked in the health field. In fact, I got 20 job offers to work at oil companies, 4 from Exxon alone. And I wasn't that good, but they would just be hiring everybody, in the early 1970s that was a chemical engineer. And all my friends went to those companies. So I initially thought I would do that too. But I remember going on these interviews, and one of them I remember at Exxon, they said, gee, if you could just increase the yield of this one petrochemical by 0.1%, they said that would be wonderful, it'd be worth billions. And I remember flying back to Boston that night thinking to myself that I really didn't want to do that. So I started thinking of other things that I might do. I was one of the things I did when I was a graduate student was start a high school school for poor high school children. And I got very involved in developing new chemistry and math curricula. And one day I saw an ad to do just that at City College of New York. So I wrote them a letter applying for a job, but they didn't write me back. But I really liked that idea. So I found all the places I could to write letters to like that. I found about 40 of them, and I wrote them all, and none of them wrote me back. So that wasn't going so well. So I started to think, what other way could I use my chemical engineering education to help people? And I thought about medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. And then one day, I walked into the lab, and one of the older scientists said to me, he said, Bob, there's a surgeon at Children's Hospital named Judah Folkman, and he sometimes hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. Anyhow, if we go to the next slide, what I'll show you is Dr. Folkman's idea. And he had this idea that if you could stop blood vessels, maybe that would be a whole new way of treating cancer. And this slide shows what he had in mind. On the lower left, you see a tumor cell somehow has become abnormal, and the cell grows in a 3-dimensional mass. But when it gets to the middle, the bottom middle, it's say has maybe a million cells, and it runs into a nutrition problem. Cells in the center can't get nutrients or get rid of waste. So it can't get bigger than that, except for the fact that he said it sent out a chemical signal called tumor angiogenesis factor. And that causes the blood vessels, which normally don't grow at all, to start growing to the tumor. And then what you see happen is on the upper right. Now the nutrition problem for the tumor is solved. It gets bigger and bigger. And if you go to the highest picture on this this upper right, what you see is it's very vascularized, and the tumors will spread through those blood vessels. That's a process called metastasis, and that's what kills. Now, his idea was that if you could stop the blood vessels, achieve anti-angiogenesis, like I say, that maybe would be a whole new way to think about treating cancer and perhaps other diseases. So how do you think about a problem like that? Well, my job was to prove he was right, and so doing, isolate the first blood vessel inhibitor. One of the things we looked at was cartilage. The next slide shows a picture of a cow bone. And what I did is we started out with little rabbits, but we couldn't get much cartilage. Then I went to cows. Chemical engineers learn scale up, but still, even one cow bone, I couldn't get that much cartilage. So I found out, where do all the bones in the northeastern United States go? And they go to these meatpacking places in South Boston. So I made an arrangement with them to get all their bones, and I'd bring them back, I'd scrape the meat off, and I'd get this white glistening cartilage that you see in the top here, and I'd slice it off and put it through various extraction and purification procedures. So that at the end of several years, I had maybe 50 to 100 fractions that I wanted to test to see if they could stop blood vessels from growing. That brings up a second issue. How do you study blood vessel growth? That's actually very hard. That's what's called a bioassay. And one of the things that made it so hard is that there's, in any test you might have, there's almost always background blood vessels, except we thought maybe for one or two places. And one of them was the eye of an animal, the cornea. And the next slide shows what Dr. Folkman and I thought about that, and ultimately would publish in Science 45 years ago. And the idea is that maybe you could have a certain type of tumor, like a V2 carcinoma, that you could place in the eye, and it will cause, as I will show you, blood vessels to grow to it over maybe an 8-week period. And then maybe we could have a slow release vehicle, like a polymer or lipid, that could slowly deliver the different macromolecules, the molecules in cartilage that we're isolating. Now, Dr. Folkman was on the board of the one company in the world working on something like this, a company called ALZA. And they had some very famous people, Nobel Prize winners in chemistry, on their board. And he asked them if they could help. But when he did, the next slide shows what they said. And they basically said you couldn't do this because the large molecules can't slowly diffuse through solid materials any more than any of us could walk through a brick wall. The literature said the same. Next slide. That the use of polymer matrices for slow release systems has been virtually restricted to small molecules. But if we were going to solve this problem of angiogenesis, we had to solve the problem of developing these delivery systems. So I spent 2 years working in the lab, experimenting with different materials, polymers, lipids, different designs, different structures. I found over 200 different ways to get this to not work. But eventually I was able to make little microspheres or nanospheres. Next slide. And you see what picture of one on the left and a cut in half on the right. And what we showed and published in Nature in 1976, next slide, was that you could use this approach to deliver molecules of any size for over 100 days. In fact, in the same paper, we even showed you could deliver nucleic acids. Nucleic acids are like DNA or messenger RNA. And this is, to my knowledge, the first time that had ever been shown you could release these from tiny particles, something I will talk about later as I go over the company I helped start called Moderna. But this was, like I said, the first time this had been done. We even showed that you could deliver molecules constantly if you wanted to. The next slide shows a picture of that. This is delivering albumin, which is 68,000 molecular weight, for over 50 days. But when I first presented this work at a scientific meeting, this was also in 1976, next slide, it was ridiculed. I remember being 27 years old and giving this talk. And when I did, I thought I did okay. I didn't forget too much what I was going to say. But when I got done, all these older scientists came up to me and told me just like what the ALZA people said. They said, what I talked about can't be right. They said, these large molecules are too big to diffuse through solid materials. And they also pointed out to me how naive I was that didn't I know that organic solvents would destroy peptides or proteins or nucleic acids. And actually, things went downhill from there. I tried to get research grants, but my first 9 were rejected. The reviewer said, how could Dr. Langer do this? He's an engineer. He doesn't know anything about oncology, even less about biology. So that wasn't so good. Then I liked being a postdoc, but a lot of my friends said it's probably not good to be a postdoc forever. So I started applying to faculty positions in my major discipline, chemical engineering. But not a single chemical engineering department in the world would hire me. So that wasn't so good either. But Dr. Folkman knew Nevin Scrimshaw, who was a very famous nutritionist at MIT. He was also on that ALSA board, and he introduced him to me. And Dr. Scrimshaw, who was offered me a job. The only thing is he was kind of department head who was like a benevolent dictator. He liked me, so he offered me the job, but he didn't bother to ask the rest of the department what they thought. Now, that might have been okay, except for one thing, which was that the year after I joined the department, Dr. Scrimshaw left. So a lot of the senior faculty decided to give me advice. And their advice is I should leave too. The next slide shows what one of my good friends at the time, still, Mike Marletta, who later became a member of the National Academy of Sciences and chair of Berkeley's chemistry department, said, reflecting back when he and I were young professors at MIT in the late 1970s. And here is a direct quote from a speech he gave. One evening, I went to a faculty dinner at a Chinese restaurant with myself and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So that was very discouraging. And you have sort of two choices. You can quit or you can keep going. And I chose the latter. And one of the things, of course, that was key since we were doing all this and everybody said it was impossible was could it possibly work? So to understand this better, one of my graduate students, Rajan Bawa, started cutting thin sections through the delivery systems. The next slide shows a picture of one of these. This was a model polymer we used, ethylene vinyl acetate. And if you didn't put a drug in it, and you took a slice, 5-micron thin section, it looked like this, just like a sheet. And molecules 300 or greater couldn't get through from one side to the other. And if that's true, then how could the molecules get out? Well, what the next slide shows is that if you put like a reddish marker drug like myoglobin in, and you cut a thin section before any release, you get what we call as a phase separation. The polymer is white and the drug is red. And then if you release these for about a year and then cut a thin section, what you see is shown on the next slide. You see all these pores that are formed where the drug was previously, and they weren't there 2 slides ago. And it's through those pores, it turns out, that the molecules get through. Now, these pores, turns out, are incredibly winding and tortuous. It takes a really, really long time for them to get through. They have tight constrictions and so forth. And over the years, our students have worked out mathematical models for predicting the release systems. But by designing them, you can get release anywhere from a few hours to many years. And since we could do this, now we could solve the angiogenesis problem. The next slide again shows the assay we wanted to develop that was published in Science. But now we had these systems, so we could put all the different fractions in and test them. And I'm going to show you a very bloody slide in a second, so I want to give you a warning. If people don't like the sight of blood, don't look. But what we did is we literally tested about 2,000 rabbit eyes. We tested 50 to 100 fractions. Almost all of them did not work. In fact, all of them but one didn't work. Those you could kind of think of as like the controls. And now I will show you the slide, the next slide. And on the left, what you see is without the cartilage-derived inhibitor. This is 8 weeks after the start of an experiment. You get the sheet of blood vessels growing right to the tumor. And if you looked at this eye 3 weeks later, it would be three-dimensional. Out of the orbit of the eye, it would actually kill. So we sacrificed the animals before then. And this happens 100% of the time. 2,000 out of 2,000 eyes. But when you put the inhibitor in, striking what happens on the right-hand side, notice that the vessels are lower. They avoid the polymer in the tumor. And it turns out that 60% of the time, these tumors never took off. So this showed for the first time that angiogenesis inhibitors did exist and provide methods by which people could isolate them in the future. Nonetheless, it took 28 years from this paper in Science before the first angiogenesis inhibitor was approved by FDA. And that was because of great work done at Genentech and other companies, and literally billions of dollars spent by pharmaceutical companies. The next slide shows what happened starting 28 years later. One after the next, angiogenesis inhibitor is approved by FDA. Avastin, the first of these, is probably the number 2 or 3 biggest biotech selling drug in history. And you see the approvals continuing. And not only are they approved for all kinds of cancers, as you can see, but also they are the only treatments, the only pharmacologic treatments for diseases of blindness in the back of the eye, like macular degeneration or diabetic retinopathy. And the drugs that treat that, like Lucentis or Eylea, they've been blockbuster drugs as well. We also thought that not only would the drug delivery work we did be useful for isolating and studying angiogenesis inhibitors, but they could be useful in their own right. An example of that, next slide, as just a proof of principle, was done by my graduate student, Larry Brown, who made an aspirin-sized pellet of insulin and designed it the right way and could lower the blood sugar with a single injection or implant for over 3 months, from 400 to 100. And we found other uses too. Well, one day, Dr. Folkman said to me, Bob, we should file for a patent. You know, that might seem very commonplace now. We have a lot of patents, but then I had no patents, and Boston Children's Hospital, where we were working, had no patents either. So we filed for a patent for the first time in the history of Children's Hospital. And 5 years in a row, the patent examiner rejected it. And so the head of the tech transfer office at that hospital came to see me one day, said, Bob, you have to stop, quit. He said, you're wasting so much money. The patent examiner will never allow this patent. He just rejects it every time. But I don't like to quit. So I started to think, how could we convince the patent examiner to allow the patent? And as I mentioned to you earlier, one of the things that when we started doing this is everybody told me it was impossible. It could never work. So I wondered, maybe somebody wrote that down. So I did what's called a science citation search. Now in 1982, looking back at our 1976 Nature paper to see who wrote about it. And I found a number of quotes, but one in particular, shown in the next slide, was, I thought, very useful. This turned out to be in 1979. I had no idea it was written, but it was by 5 of the top material scientists in the world. And they wrote, describing this young field, that generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising, so surprising is a really good word for a patent examiner, results that clearly demonstrate the opposite. So I showed this to the head of the tech transfer office. He flew down to Washington to show it to the examiner. And the patent examiner said, I had no idea that this was true. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from each of these 5 people that they really wrote this. So I wrote each of them, and they were all nice enough to write me back that they did write it. And then the patent examiner allowed the patent. The next slide shows that. It was controlled release of macromolecules. And with that patent, we would start licensing it to different companies, teaching different companies. I got involved in starting different companies. And today there are many, many products based on this. The next slide just shows some pictures. And all these have been developed by different companies, but basically they're little microspheres like Lupron Depot that can put a peptide in it. And normally the peptide wouldn't last more than a second or two. And of course you can't give it orally or transdermally, it's just too big. But when you inject it, now it works for 1 month initially, now even over 6 months. And there are many others new on this slide, new ways of treating opioid addiction, pain, type 2 diabetes, different eye diseases, schizophrenia, and so forth. And we want to take this further. You know, so all these pictures, they're all injected or implanted, but underneath the skin. But what if you could actually even target something right to the entity, like a tumor? So we thought about making these, and we could do this, we'd even done it, made them even smaller, like nanoparticles. But when you do that, a lot of times they get eaten by macrophages. So we made these coatings. The next slide shows a picture and of what Roxandra Greff, when she was in our lab, did. This was a paper we published in Science almost 30 years ago. But our idea was that if we coated these little nanoparticles with polyethylene glycol, then that could provide a disguise and they would last longer. And we could even take this further. And on the next slide, you could even put targeting molecules in. This was done by Omid Farrokhzad when he was in our lab. And let me just show you a video that Nova, the TV show, did of our work, because they explain it probably a lot better than me. If we could go to the next slide or video.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
Now, I should say that a lot of the clinicians I work with tell me it doesn't really blow the cell up that way, but you get the idea. And you could use this for small molecules, which we're doing. But what's really exciting and what I wanted to turn to now is that it really permits genetic therapy, like delivering siRNA and messenger RNA. In fact, one company I was on the founding scientific advisory board is called Alnylam. If we could go to the next slide. And one of my students, Kenna Kink, led a lot of this effort, but they've developed siRNA therapeutics. But if you just injected them, generally they get destroyed. But what Akin and the team did, and I was fortunate to be able to help a little bit, is put them in little lipid nanoparticles in this case. Again, they have polyethylene glycol coatings on them and other components. But when you do this, that permitted really the first siRNA therapy. And that's for treating ATTR amyloidosis. And that's now been in use since 2018. That's called Onpattro. But let me now go back to 2010. Next slide. And another thought that we had, this was work I'd done with Derek Rossi, Ken Chen, and Nubar Fayan. But we had this idea that maybe we could make messenger RNA therapeutics. You may know the central dogma of molecular biology is DNA makes RNA makes protein. And of course, protein therapeutics have become enormous, hundreds of billions of dollars of sales a year. And also we thought that you could treat new diseases that you could never treat before, like intracellular diseases or and so forth, where there's problems with the protein. Also, protein therapeutics take a long time to make and involve extensive purification and production. Whereas if you could ever do messenger RNA, that would be much faster because the body would do all the work. If you could make the messenger RNA, say put it in a nanoparticle, inject it into the body, it would make the protein. So you could actually come up with ways to make it much faster, and I'll give an example. So in 2010, we started this company called Moderna. And of course, just to go over an example of speed, Moderna actually has 14 products in clinical trials. But the example, of course, and you may all know this, is the COVID vaccine. And the next slide just shows what Moderna, the great scientists there, did. I'm still on the board of directors and on the scientific advisory board. But so I have obvious conflicts, but these are facts, what I'm going to tell you. In January 11, 2020, the Chinese scientists published the genetic sequence. Just 2 days later, at Moderna, they finalized the messenger RNA vaccine design. Within about a month, the first batches in the lipid nanoparticles were shipped to NIH for testing. And the first clinical trials were done, less than a month from then. And of course, by the end of the year, it was approved for emergency use authorization all over the world. And nonetheless, I think it's important to realize that when we started this, now it seems like obvious, or everybody says, sure work. But that wasn't the case when we started it. Moderna and actually also BioNTech, Pfizer, also using nanoparticles with polyethylene glycol on them. Everybody was criticizing them, and all kinds of supposed experts said this wouldn't work. Just to show you one example, next slide. Our local paper, the Boston Globe, pretty well-known paper, and this is from May of 19th, 2020, not very long ago. They interviewed a lot of supposed experts, and they kept saying, this isn't how you do science. This is front page headlines with my picture on the front page. Wasn't very excited about this, but that's the world we live in. And of course, as we show on the next slide, you do see the Moderna vaccine, and fortunately, it's been able to help people all over the world. I wanted and I think there's a lot more to come. I mean, other vaccines, other kinds of therapeutics, you know, I think it will change medicine. And again, delivery using these nanoparticles to protect the messenger RNA made it possible. Another thing that we've been interested in, I'm going to shift gears a little bit, is oral delivery of complex molecules. And that's not so easy. If you try to deliver a large molecule orally, if we could go to the next slide, it has a tough journey. When you swallow something, it gets hit with stomach acid, it gets hit with proteases, and then finally you got to get through the intestinal wall, and that really doesn't happen very easily. But if we could go to the next slide, what people have done is that they can do injections. But all those injections into the gut are always come through your rear end. You do some type of endoscopy, and that actually works. It's got high delivery efficiency, very little pain because of the low amount of nerves in the stomach, and the tissue regenerates rapidly. So that's great, but I don't think any of us necessarily want to have injections routinely through our rear end. But we were thinking with Gio Traverso, one of my fellows at the time, now professor at MIT and Harvard, that maybe what we could do is come up with a way to swallow something and inject it. So how could you do that? So what we came up with, and we've actually designed a little pill to do that, is it's got a number of features. Basically, I'll just show you. It's this small, and you could put it in a capsule. But what we've done is there's this little hole, and you've got behind that hole a needle. But that needle is going to be made out of almost entirely whatever drug you want, let's say insulin. Behind that needle, you have a spring, which, if cocked, will release the needle and shoot it right through this little hole, and it'll go into the gut. But of course, you don't want it to happen right away, right? You want it to maybe happen 30 minutes to an hour later when this pill is now in your stomach. So what we did is we embedded the spring in a sugar glass. And so the idea of the sugar glasses, and I'll go over this, is by controlling its thickness, we can make it break whenever we want. So we can program that in, because it's going to break by osmosis 30 to 60 minutes, let's say 30 minutes. But you might be thinking one more thing. How do we make sure that this where you have the hole is always going to be facing the gut wall? So Gio and Alex in our lab, basically what we did is we modeled this like a leopard tortoise. And I'm just going to show you what we did. But the beauty of this is that it's weighted in such a way that no matter what you do, it will always land with this face down. If we could go to the next slide. So that's just a picture of what happens. Because of the weight, and I could actually do this myself, no matter what I do, it'll always land like this. So at any rate, and as you can see on the right-hand side, because that does, now it's self-oriented, and when it gets there, it will shoot the drug out, as you can see from the red part right there. And just to show you a little bit more about the self-orientation, the next slide gives you the idea of the leopard tortoise. It just always will turn over until it self-orients. You could do it 1,000 times out of 1,000, and it'll just shoot it out like this. And I mentioned to you also that we have this spring behind the needle. And like I said, the needle's made all of the drug, but the spring, depending how thick you make it, will break at whatever time you program into it. And the next slide just shows a picture of the diameter with the time of actuation. We can control that time by the diameter. And this is just a picture, a video of the spring just shooting out. And when the spring, of course, the spring can't get out of the device, but the needle will because of the size. And what the next slide shows you is that perforation risk is mitigated by controlling actuation force. In fact, if you look on the right-hand side, you see histology, and there's really no tissue damage. Also, on the next slide, we've shown that this insulin is very stable, much more stable than liquids. And finally, on the next two slides, we show that it actually works in an animal model. This was all published in the journal Science. And what you can see on the left-hand side is that basically you get similar insulin plasma levels to subcutaneous injections. And on the right-hand side, again, really no tissue damage. And not only do you get the same insulin levels, this is all pigs, but on the next slide, we show that you get the same lowering of glucose. And again, no tissue damage. So here we've gotten funding from and have worked closely with the company Novo Nordisk. And they are bringing this into clinical trials, in the next few months, as my understanding. And of course, not only could it be useful for delivering insulin someday, but we've published papers in various Nature and Science journals that it could be useful for delivering antibodies, RNA drugs, all kinds of things. So all kinds of macromolecules. So we're very excited that this could provide someday perhaps a new paradigm. I want to now turn to new materials. And let me just go over on the next slide how materials generally found their way into medicine. Almost always they were found by medical doctors going to their house and finding an object that they wanted to, at their house that they wanted to use. So the artificial heart, for example, were clinicians saying, well, what do I really want in an artificial heart? And they said something with a good flex life. So they said, what object has that? And they said a lady's girdle. So they asked, well, what is that made out of? Well, it's made out of a polyether-urethane. So that's what they made the artificial heart out of. So, and that's still what it's used today. In fact, that was started in 1967. But the artificial heart has some problems. When people put the artificial heart in a patient, blood can hit the surface of the heart and get a clot, because it's an unnatural substance. The clot goes to the brain, the patient gets a stroke and they die. Yet something that's designed to be lady's girdle might not be the optimal blood contacting material. So, and this problem pervades all of medicine. Breast implants, for example, one of those was a mattress stuffing because they wanted something squishy. So being a chemical engineer, I thought, well, maybe we could try a very different approach, which is rather than taking something from a house, what if we ask the question, what do you really want in a biomaterial from a chemistry standpoint, engineering standpoint, and biology standpoint, and then design it from first principles? So we picked an example. Next slide. And that example was synthetic degradable materials. And when we started, the only materials that were approved by FDA were the polyester sutures, and they dissolve like this, as you see here. And when you look at this, it starts out like this phone book structure, the drug's uniformly distributed. Then if you go to the second panel, it gets spongy, and the third panel, it falls apart, which is okay for some drugs, but for others like insulin or an anti-cancer drug, you get bursts of drug coming out, and that could be fatal. So we said, from an engineering standpoint, what do you want? Next slide. We said surface erosion, like a bar of soap dissolves. And how do you do that? Well, we then asked different design questions, like what should cause the degradation? Should it be enzymes, or should it be water, or something else? And our thinking was, everybody has excess water, so let's build in something that will degrade by water. And then we went further. I got very good chemists and biologists to help us, and we figured out the right monomers, the right bonds. And the next slide shows what we wrote on the blackboard. This is a polyanhydride that is a copolymer with these two hydrophobic units designed to keep water out. So you get surface erosion. But by changing the ratio, our theory was that you could get different erosion rates. So to test that out, we made different amounts of each of those two units. Next slide. And what you see is that, in fact, is true. 79% of the one unit, it's all gone in 2 weeks. This is only a millimeter thick system. Yet 0% of that unit, sebacic acid, it'll take 3 or 4 years for this to dissolve. So you could simply dial in the ratio of these monomers, and you'll get any erosion or release rate you want. And with that in mind, we thought, well, there's diseases we'd like to treat, like brain cancer. Next slide. This was brought to us by Henry Brem, a young neurosurgeon at Johns Hopkins, who's now chief of neurosurgery there. And the disease that he wanted to treat was glioblastoma multiforme. And it's a uniformly fatal disease. And even if you hit it with everything, surgery, radiation, and chemotherapy, the mean lifespan was 50 weeks. And the drugs that they use, next slide, this is when we started in the 80s, the only one was BCNU. And it's an awful drug. It travels throughout the whole body, causing side effects in the liver, kidney, and spleen. But what Henry and I talked about is shown on the next slide. Maybe we could develop a new paradigm, local chemotherapy. Could we allow the neurosurgeon like Dr. Brem to operate on the patient, which he's going to do anyhow, but then before closing up the patient, line the surgical cavity with polymer containing this BCNU, little wafers. Now, normally, this drug has a 12-minute lifetime, but the polymer will protect it from degradation. And the beauty of this, theoretically, is you get high concentrations in the brain where you want it, and low concentrations in the rest of the body where it could cause harm. Now, when we first proposed this, we had to raise money for it. And so we write grants to the federal government, and we did terribly. I saved a lot of the reviews. The next slide shows some of them. When we first wrote this, the reviewer said the polymers couldn't be synthesized, but I had a very good graduate student named Howie Rosen, and he was able to synthesize them. He later became president of Alza Corporation, a $12 billion company, and was elected to National Academy of Engineering. Then the reviewer said, well, okay, that the polymers have anhydride bonds, they'll react with whatever drug you put in. But I had another couple postdocs, Cam Leung, who's now a chaired professor of biomedical engineering at Columbia, and Bob Linhart, who's a chaired professor at RPI, and they showed that this didn't happen. And then we sent it back again. The reviewer said, well, the polymers are low molecular weight, they're fragile, they'll break. But I had another couple of postdocs, Avi Dome, who's now dean of pharmacy at Hebrew University, and Edith Mathiowicz, who's a full professor at Brown University, showed you could make polymers very strong, so that didn't happen. And then we sent that back again, and the reviewer said, well, but these are going to be toxic. But I had another graduate student, Kata Lorenzen, who actually received the National Medal of Technology from President Obama, one of the highest honors in the United States. He also became Dean of Medicine at University of Connecticut, and he showed that they were actually very safe. Then we said, and then the reviewer said, but the drug won't diffuse far enough. But Mark Saltzman, who Yale University recruited to be their inaugural department head for their first department of biomedical engineering, and he's been elected to multiple national academies, he showed that it did diffuse far enough. Anyhow, this kept going and going on and on until 1996 when the FDA approved it. You can probably tell from the way I'm talking that I'm very proud of how well all the MIT people did. They got great jobs, great honors, whereas the reviewers, they didn't do very well. Let me just show you what this looks like. I'm going to show you some human pictures, so they're going to be bloody again, but these are brains that patients that Henry Brem treated. There's a little wafer going into the brain. Next slide. Usually they put 6 or 7 in and close up the brain. Next slide. I don't usually like to keep those on very long. In fact, I remember one time I gave a lecture at MIT a number of years ago, and my wife came, and at the end of it, I asked her, I said, well, Laura, what did you think of the lecture? And she said, well, Bob, the lecture was okay, but you had those 2 bloody slides on for 10 minutes. And she said, I don't know if you were looking, but all those poor engineers were turning green. So ever after that, I did what I did today. I gave a warning, and I left them, and I put them on for a couple seconds. But just to tell you the sequel to that was a few years later, I got asked to give this dinner speech. This was actually to a group of neurosurgeons. And when I got done, when I got to this, I said the same thing. But at the end of the talk, a bunch of the neurosurgeons came up to me and they said, you know, those 2 bloody slides you showed? And I said, yes. They said, those were fine. They said, no problem. But they said, those chemical formulas, I mean, to leave those on for a neurosurgeon, they said, that's awful. So now I kind of adjust what I do depending on who I'm talking to. So this is just some of the clinical data that this was from one of the first trials in Sweden and Finland, where you see that this is not a cure, but it does prolong life. You see 5-fold improvement at the end of 2 years. The next slide is a more recent one where people looked at over 60 trials, 5,000 patients, and again shows that this is effective. Again, not a cure, but this is now has been used for the last 25 years in over 30 countries for treating glioblastoma. And equally, if not more importantly, it created a whole new paradigm of local chemotherapy, which is not only now used in certain kinds of cancers, but is probably one of the predominant treatments for treating heart disease. And let me just show you that on the next slide, which is what's called the drug-eluting stent. And today, if somebody has heart disease, one of the most common treatments is to put this stent in, which is almost like a Chinese finger puzzle, and it keeps the blood vessel open. But within 6 months, maybe about 50% of the time, because of smooth muscle cell proliferation, the vessel closes. So what now is done is to take a different anti-cancer drug like Taxol, put it, you don't want to give it systemically because it'll cause toxicity at the amounts that are needed, but you can deliver it locally by making a nice thin coating on the stent, and it will keep the blood vessels open. And these are used by millions of patients every year. So I thought I'd end the talk by telling you about one other thing that I think is exciting, where materials can help people, and that's to create new tissues and organs. And let me motivate that on the next slide by showing you a picture of a little baby who's dying of liver failure. He was one of the patients of Jay Vacanti, who was my collaborator in this. And the only way a child like that would live is somebody else would die. And Jay or whoever is the patient's doctor would do an operation to transplant a liver. But there's not nearly enough to go around. So Jay asked me, must have been around 1981, could we make new tissues and organs from scratch? So we came up with the following idea. Next slide. This was actually from a paper he and I wrote in Science. It's been cited over 8,000 times. And it's just a paradigm for how you could think about creating new tissues and organs, that you could take any cell type shown on the left. In fact, you could use stem cells, but we wrote this before people talked about those. If you inject them at random, nothing happens. But the cells are smart. If you put them on a scaffold, they can reorganize. And we don't want the scaffold there forever because it may cause problems. You want it to degrade, but it can provide a template for the cells to reorganize. And then you could grow the cells in vitro tissue culture in what we call a bioreactor, and then make the tissue in the body. The next slide just shows some pictures of some of the scanning electron micrographs where you see polymer fibers with liver cells on it. And the next slide shows a vision of where we feel this might go someday, that you could use what are called CAD-CAM techniques, computer-aided design, to make tissues of organs in any shape you want. Here's a nose. This is what I'm going to tell you is pure speculation. But let's say 40 years from now, someone comes to a plastic surgeon, they don't like their nose, they want a new nose. My speculation is you'll have a computer screen with pictures of 40 noses and you take your pick. Well, what you might envision is then somebody could have a regular nose, like you see on the left. They could have a upturned nose, which you could do by taking a little bit off of the bottom of, or you could even have a hook nose. I mean, you probably wouldn't want that, but I mean, you could make anything you want. And the idea is you could use CAD-CAM techniques, computer-aided design, to make these noses of any shape, and then follow it up with techniques like foaming techniques or three-dimensional printing to make the noses of any shape. And what you see is on the right-hand side is cells have been taken out and put on the nose, and you see it, and you see that after 2 weeks in culture. And so the Vacantis and Lisa Fried and Gordana Vanyak have worked on these and made tissues from scratch. And the next slide just shows an example of that. There's nude mice, and what you can see is if you go to the animal on the bottom, what happens is that we've redone his skull. If you go to the next slide and look at the animal on the top, we've redone his cheek. If you go to the next slide and open up the animals and look at the tissue, it's pure white glistening cartilage, and actually histologically, it's also cartilage. Well, the Army came to see us once and asked Linda Griffith, who was one of my postdocs, now a professor at MIT, Jay Vacanti and myself, they said, well, we have soldiers coming back from Iraq and Afghanistan. Could you make new body parts, like new ears for them? So Linda, next slide, created a human ear from a scaffold. And what you see on the bottom right, the top left is this ear. On the bottom right is a high-powered scanning electron micrograph of polymer fibers, which will ultimately dissolve into water and carbon dioxide. But as you look at the bottom part of it, you can see cells and extracellular matrix growing. And ultimately, it will make an ear. And just to show you that, the next slide shows the human ear on the rabbit. Now, if I go, I'll again go to the next slide. The reason you want to do that is, of course, to show that it's safe, which it was shown. But what that opens the door for is treating patients with different problems. Not only the soldiers, but here's a 12-year-old boy who was the patient of Jay Vacanti, who was my collaborator. But look at this 12-year-old boy. He's got no chest covering his heart. Like other 12-year-old boys in the United States, he likes to play baseball. But if he were ever hit in the chest with a baseball, he could die. So Jay operated on him. We took his cells. We made a polymer scaffold, and Jay made a new chest for him. And now he's 6 feet tall and doing well. We also licensed this technology to different companies. One in particular, several actually could make artificial skin for burn victims or patients with diabetic skin ulcers. And let me show you that. Next slide. So this is a 2-year-old boy who's very badly burned. But you could take the product, which are neonatal skin fibroblasts, which could be even cryopreserved on the polymer scaffold. Next slide. You could take that and put it in the child at the time of injury. Next slide. Let's come back 3 weeks later. And now let's come back 6 months later. So these are now FDA approved. They're widely used for burn victims and patients who have diabetic skin ulcers. But also there's a lot of other tissues that myself and our lab and our collaborators, a lot of my former students have been developing. So Laura Nicholson, for example, started a company called Humacyte, which is in advanced phase 3 trials for creating new blood vessels. David Lucchino and Chris Luce, 2 of my students, they've worked out, and Jeff Karp, they've worked out strategies for hopefully treating hearing loss that's in clinical trials. Others have developed ways to treat spinal cord repair, diabetes, and other diseases. So the idea is that you could use this for hopefully creating new tissues and organs. You could also use it for delivery systems. You could genetically engineer these cells to produce different types of molecules, like erythropoietin or factor VIII. In fact, Sigalon, a company we started, is with Dan Anderson, another of my former postdocs, now professor at MIT, is doing that. The last thing I wanted to say, though, is that not only might you envision creating new tissues and organs for patients, but right now, whenever a pharmaceutical company does drug testing to see if it's safe and effective, we use animal models and we use humans. But maybe what you could do someday to reduce that is create, and to speed it up even, is to create organs or tissues on a chip. And I thought I'd just end with an example of that was done by 2 of my other students, led by Gordana Vanyak, who's now professor at Columbia. And there's a company called Tara Biosystems that I've been on the board of. And so what they're doing is actually creating hearts on a chip. And this again goes back to work that happened when they were in our lab. Next slide. You could take heart muscle cells, put them on a scaffold, and when you do, you get this characteristic cardiac-specific ultrastructure of sarcomers and intercalated discs. And I'm even going to show you a video of them. And this is 6 million cells, and they're beating as one. We're not shaking this or anything. It's just beating like a heart. It's really remarkable. It's just, you look at a microscope and it's just beating like a heart. And you can even take an EKG of it. Next slide. Actually, that's the last slide. But and so you can take an EKG of it, and it's just like doing a regular heart. Now, what they're doing at Tara Biosystems is is doing drug testing with many companies, I think close to 30 companies. And the idea is you can accelerate testing by doing high throughput screening, since you can test a lot of these quickly in vitro, and you can look and see whether the drugs are safe or whether they're toxic or whether they're effective. And so this could greatly someday minimize animal testing and human testing. And this is hardly the only example. Gio Traverso, who was I mentioned before, who's now professor of gastroenterology at Harvard and mechanical engineering at MIT, and our colleagues, they've actually developed a gastrointestinal tract on a chip. And it's far, far more effective at looking at drug absorption than CACO2 cells, which has been the gold standard for 50 years. Here, which we published in Nature Biomedical Engineering, is that you get pretty much 100% correlation with this tissue on a chip compared to the CACO2 cells. And others like Linda Griffith and Sangeeta Bhatia have created livers on a chip. Ourselves, this is still an early stage, we're working with Li Wei Sai at MIT to create a brain on a chip. But all these things may someday accelerate animal testing and human testing and so forth. I guess the last thing I thought I'd say is there's applications of this that never occurred to me when we started this. Shulamit Levenberg, for example, another one of my former postdocs, who later became dean of biomedical engineering at Technion, she's actually using these approaches to make meat in addition to everything else. I think they've raised several $100 million to make meat this way. Ali Katamasani, another one of my students, is doing that too, as are others. And then there's even companies making leather from this. So it ends up, as many things do when you do science, one application or one discovery leads to the next. And I'll end that way. I mean, to me, I think even though I've tried to go over a number of these things that you could do by combining engineering with, with chemistry and biology, that there's so many things that we'd like to do, so many new treatments that I hope will come about in the future. But that being said, I'm thrilled to see that drug delivery and pharmaceutical technology has made a difference in so many diseases, including the whole COVID problem. And I hope that tissue engineering will also end up, and I think it's off to a good start, also making a big impact in the pharmaceutical area. Thank you so much for having me.
I think he's supposed to be joining us. Is he online?
I'm here.
Oh, fantastic. Thank you very much. Hello, Professor Langer. Thank you very much for joining us. We just heard your talk. Great talk. Highlights the impactful work that you're undertaking, also highlights the importance of interdisciplinary research. So are you okay to take some questions?
Absolutely. That's why I'm here. It's an honor really to be presenting to you and to be able to, be with you at least virtually today. Thank you so much for this honor.
No, and I apologize that, I asked you a while ago and you were supposed to come to the UK and we had to cancel you several times to come and give this talk and so on, but thank you for doing it today.
Oh, well, the circumstances certainly warranted it. No apologies.
It's definitely worth waiting for. Thank you. So maybe I can start off with a question while people may be thinking about questions and so on. Your title says developing solutions for global challenges. Some of the technologies you're developing may be relatively expensive and may not be affordable by developing countries and so on. I don't know if maybe your perspective on that, how we can actually develop technologies that that make it available for the rest of the world rather than just the rich countries.
Yes, well, I think that's a great question. And in fact, I'd say half of what our lab does, and again, there was only so much time to put things into the talk, but about half of what our lab does is exactly on that issue. We've been working very closely with the Gates Foundation and actually Bill Gates himself on several things. And I'll just highlight them, all of which actually involve some of the things I talked about. But one, our super long acting oral delivery systems that somebody could take So you'd only need to take the drug once. In fact, we're working on that with the Gates Foundation for malaria and TB. And of course, if you just had to take one pill, then obviously the cost would go way down, and actually patient compliance would go way up. We've also, when we published some of this in Science, developed what I'll call self-boosting vaccines, so that you can just give, ideally, again, this is still in the research stage, but it's a new printing process where you could actually create a cocktail of little micro or nanoparticles that would, you injected at time 0, but then you get bursts at 1 week, 1 month, 2 months, 5 months, a year, and so forth. And actually what we're working on now is actually putting them in little microneedle patches that you could distribute inexpensively, but also much more easily. It's like just putting a Band-Aid on. And we're doing that, including for messenger RNA. And then finally, we're also working on, and again, this is more out of the drug realm, but there's about 2 billion people in the underdeveloped world that have nutrition problems. And so we've been developing new ways of encapsulating very sensitive micronutrients that a lot of these children and adults don't get enough of, like iron and vitamin A, in ways that they can withstand boiling and high temperatures and light and so forth. And some of this we've actually published in multiple journals, like Science, Science, Translational Medicine, and others. But we have probably 80 people in our lab working on these projects. So I'm actually very glad you asked the question. I thought about putting that in the talk, but I wanted to highlight, sort of the breadth.
Great. Thank you very much. Really great to hear that. And so on. Any questions from colleagues in the audience? Maybe I can ask you another one if that's okay.
Anything you want.
So the mRNA platform, Moderna, and obviously Pfizer as well has got the mRNA platform. Been great success, billions of doses of vaccine being given and so on. I just want to hear your perspective on how the mRNA platform may be used in the future for other therapeutic areas and so on, particularly cancer and so on.
Yeah, well, also great question. And I mean, maybe I'll just, and this is all public information, I just highlight Moderna's platform, and Moderna's actually in over 14 clinical trials, and actually personalized cancer vaccines are one of those areas. Of course, when you're talking about COVID, it's a prophylactic, right? You're trying to prevent something. In the case of cancer, somebody already has it, so you're doing what I'll call a therapeutic vaccine. And the idea, and that's actually why Moderna, we built the manufacturing plant in Norwood near Boston in the first place, was to do just that. And the idea is that you could someday, and we're actually, like I say, in early stage clinical trials, take a portion of the patient's tumor, characterize it from a genetic standpoint, and then maybe direct 24 or 36 messenger RNAs just against some of these epitopes, so to speak. So that's on cancer. But then there are many other vaccines, including flu, our respiratory viruses. We're in clinical trials with AstraZeneca for heart disease to create new blood vessels. We're working with Vertex on new treatments for cystic fibrosis. I think messenger RNA, is a very broad platform. And of course, other companies are like not just Moderna, but BioNTech and Pfizer and now Sanofi, working on this too.
So do you think that we may have a vaccine in the future which may be multivalent, that you can actually have respiratory syncytial virus, flu, coronaviruses and other things which you actually be given together at the same time using the kind of mRNA platform that you're talking about?
I think that's exactly right what you said. That certainly is one of the plans that we're looking at.
Great. Please, Humphrey.
One of the problems that must immediately arise with these ultra-long-acting polymer release systems, which go on releasing drug for years on end, maybe, must be the problem of developing side effects, and you need to reverse them. You need to stop the drug from coming out. Have you got any ideas about terminating the action?
Yeah, well, that's an interesting question. Of course, for some of them, they've had, long histories of being used, without terrible side effects. But your question's really a good one as we think about new things. So the answer to that is yes. I mean, for degradable ones, so what we've done are potentially degradable ones, either orally or injectable ones. There are ways that you could orally, with the strategy we've developed, give something that you could swallow, and it'll basically break up the system in sort of pieces that will ultimately get excreted. For injectable ones, a lot might depend on what you're doing. I mean, there are ways that you could retrieve things. We've published papers, for example, where we've put tiny little safe magnetic particles in these, where there are ways to remove them. And we published one of those in Proceedings of the National Academy of Sciences. So it's an interesting question. Probably it would be done on a case-by-case basis, given what route of administration you're thinking about, what the negative consequences, as you're pointing out, might be, and so forth.
Thank you. So I've got a question online. This is a hybrid conference, and we've got about 200 people online. And the question is that, are you working on technology which allows you to be able to silence overexpressed genes? And can you give an example of a particular disease where you're doing that?
Well, we've been doing more general work. So we actually have done work on and published some of it with Dan Anderson, who was one of my former postdocs and now a professor at MIT, where we've silenced up to 20 genes at once using RNAi and nanoparticles. What the disease might be is like our work, like I say, in the labs is more basic. Alnylam, which is the company that I work most closely with, of course, is doing work on these things. But the issue, just to go over why I should say they probably haven't picked that first as something to do, is the regulatory issues when you try to, FDA type issues, when you try to silence 20 genes at once, they may not be as straightforward as when you just try to silence one. So I think silencing one was hard enough. People didn't think that would be so easy to do. Now that's gotten 4 of those have been approved, which is very exciting. But someday I hope we will be able to do exactly what you said. But I think you build up your confidence and the regulatory, confidence and so forth before we do that. But it's a great question, and it'd be great to be able to do it. And like I say, scientifically, we have done it.
Great. Don't know if there are any more questions. Please, at the back there. This is from Dr. Pauline Williams from GSK.
Thank you. My question is about research culture, and in your talk you described multiple setbacks early on and great degree of attrition and naysayers. And I wondered if you were starting out on the journey now in your research career, whether you feel the current culture would be more enabling or less.
Well, that's a very interesting question. I think, sadly, I'd say the same. Really, I think, and all of you are scientists, and obviously are. I just think that there are always going to be a lot of naysayers. I think that's just a sad commentary on. I wish scientists were more positive. I know when I've written, grants, even now, to government agencies, and you have reviewers from different universities, they really focus on why something won't work rather than the benefits of that could happen if they do. And the newspapers, I think probably in the United States, they're just negative all the time. It's just sad, especially on the kinds of things we're talking about. They're just over and over again negative on vaccines and so forth. I hope the British newspapers are much better.
No. The answer to that is no.
Okay.
Okay, well, thank you very much, Professor Lang and Bob. Thank you very much for giving the President's lecture today for the British Pharmacological Society. Hopefully we can invite you again in the future to be able to give a lecture in person. So thank you very much for joining us today.
Well, thank you.